everyone, and welcome to Tête-a-Tech. For those of you joining us for the first time, from day one at Bay Brazil, our goal has been to connect tech players from Brazil and Silicon Valley and promote dialogue, knowledge sharing, and more opportunities within the innovation ecosystem of both regions. Today, we are having a special discussion because of course, the COVID-19 pandemic is now global. And as of today, we had 3.6 million cases around the world, 254,000 deaths. So this is a concern. Some countries are now in the process of easing the coronavirus regulations that they put in place. In Hong Kong, most workers are returning to their offices. But in some regions, like Brazil, the COVID-19 pandemic is at full force. To help us understand exactly what are the treatments that right now have been developing, or the research that has been done to de-escalate and de-accelerate the COVID-19 pandemic, we invited two scientists who are joining us today, one from San Francisco and one from Menlo Park. So I want to welcome them. Their names are Dr. Fernanda Tovar-Moll, President of the DOR Institute for Research and Education. Welcome, Fernanda.
Thank you.
And Dr. Navan Krogan, Professor at UC San Francisco and QBI Coronavirus Research Group Director. I'm very happy to have them here to give us a perspective of what's going on in both countries. And to moderate this conversation, we invited our board member, Fred Aslan, who is an entrepreneur and also a president at Vividian Therapeutics. Welcome, everybody. Feel free to send your questions here over the chat. We'll do our best to have to send them to our panelists and have them answer. Fred, I pass the ball to you.
Thank you, Margarise. It's a real pleasure to be here speaking with these two guests. Just to give all the listeners a sense of kind of the structure of this conversation, I thought we would start by hearing a little bit from you guys in terms of your backgrounds and your respective institutions. Then, Fernanda, I'm going to take the opportunity to ask you about COVID in Brazil, right? Just kind of what you're seeing on the ground and the realities there compared to what's happening in the US. It's nice because you live in the US, so you have an experience of what's happening here. It'll be useful for you to make some comparisons with what you're seeing in Brazil. And then I'd like to ask both of you, how you guys have been involved in actually looking for solutions for COVID and the scope of what those activities are. Is that okay? So why don't I pass it to you, Fernanda? Maybe you can start with the background, and then I'll jump next to Nevin.
Okay, thank you. Thank you very much. Thank you, Margarise and Bay Brazil, for the opportunity to be here. So my name is Fernanda Tovar-Moll. I am MD by training. I'm a radiologist and I'm a neuroscientist. And I work at the DOR Institute for Research and Education. The DOR Institute is a not-for-profit research institute that we founded around 10 years ago. And the main maintainer is the DOR Hospitals Network. Is the largest hospital network in Brazil, around 50 hospitals. And our mission at IDOR is to support science, education, and innovation, not only related to the clinics and the hospitals itself, but also in Brazil and Latin America, and with a lot of connections internationally also. So there's a little bit of what we're doing there.
Thank you, Fernanda. And while you're introducing yourself, maybe just to give an example to people about the type of work that your institute does. I was very curious about the work that you guys did with Zika. So would you mind just telling us a little bit about that work and the impact it had?
Yeah, so yeah, we are not a big institute. We are around 50 researchers, and but we have some areas of expertise. Neuroscience is one of them. Translation neuroscience with stem cells and neuro organoids models. Also intensive care and others more related to clinical basis. When we actually had the Zika epidemics around 3 years ago, a little bit more, what we did was actually to try to use our knowledge and our expertise to face a new problem. And I am, for example, I work with brain imaging in developing brains. But I've never worked with Zika before. So what I tried to do was to use the tools that we had to be able to describe all the characteristics that we were found in microcephaly kids and describing all the correlation with the congenital Zika syndrome, as well as our colleague, Dr. Steven Haynes, that worked with brain organoids, was the first one actually to be able to show that human cells were infected by the Zika virus. So those are one of the contributions that we with actually not, it was a very specific moment because the collaboration was very, very important. So between public and private sectors and also public, private science to be able to give a really rapid and urgent answer has to be collaborative. And I think that's what was happening in Brazil at that time with the Zika.
No, that's great. And I think Zika is one of, if you lived in Brazil at the time, you know how badly that morbidity was. And that's a pretty good success story in the sense that at least we managed to get over the peak. Is it still, just to close off Zika, because I know this is a COVID discussion, is it basically dormant right now and one will expect it one day to come back, but is it pretty dormant right now?
Yeah, it is. And actually, one thing that we learned is that we need to be fast and to organize how we could collect the data to be able to really have better response and other answers. Of course, the work with Zika is still ongoing because we have so many questions that came from the beginning, but the cases were really we had the peak and then we didn't have the case. So the majority of the clinical studies that we would have more controlled studies, actually we didn't have, and we still have a lot of questions because of that. And something that we also learn to be able how to deal with COVID, I guess. Yeah.
Thank you. And Nevin, please, would you mind giving us some background?
Sure. Thank you very much for inviting me here. I'm delighted to be here. So I am a professor and a director of the Quantitative Biosciences Institute at the University of California in San Francisco. I'm also a senior investigator at the affiliated Gladstone Institute in San Francisco. And the Institute, the Quantitative Biosciences Institute, which I'm director of, many of the labs are focused on technology development, as is my lab. So we're involved in creating and utilizing technology which allows for the unbiased quantitative characterization of molecular information of various kinds in a very disease agnostic way. And in the past, we've facilitated collaborations between the scientists, focused on different aspects of the technology, trying to put it all together and focus this pipeline on a variety of different disease areas, including cancer, neuropsychiatric disorders, and we have a big component focused on infectious disease. And so a lot of our work, we're really looking at the proteins of these different disease states and how how proteins interact with other proteins in healthy situations and disease situations. In the last couple of months, we've turned our technologies to study SARS-CoV-2. In the past, we'd focused on many other viruses like HIV, Ebola, Zika, and dengue. We've done a lot of work on Zika in the past as well, which I can briefly mention in just a minute. But we got the sequence for SARS-CoV-2 in January, cloned out each one of these genes one at a time, I think we're the first group to clone them all out, all approximately 30 genes. And we use these genes to generate a map or a blueprint of how we think the SARS-2 comes in and hijacks and rewires the cell during the course of infection. And during this project, we identified over 300 human proteins that we think the virus needs. And then we cross-reference that list to drugs and compounds that are known to target those particular human proteins for other diseases, obviously. And then in an attempt to carry out drug repurposing, we collaborated with great chemical biologists at San Francisco, including Kevon Shokat and Brian Shoykat, identified about 70 drugs or compounds that we think could have antiviral activities. And then through collaborations with a virologist in New York and in Paris, we've been testing those over the last couple of months, and we've honed in on a couple of classes that we're very excited about that have strong antiviral activities. And we're now trying to figure out how to get this information to translate this data into patients and into the clinical setting right now. So I would say we're focused on technology, and we can focus on a variety of different disease areas, and we're in a perfect position to use our technology in a very integrative way to study this particular virus, and in a very speedy and unprecedented collaborative way, I would say, bringing together scientists not just in the Bay Area but around the world as well. Great.
Well, I have lots of follow-up questions. So why don't we at least start by just discussing COVID at a high level, right? The impact that it has had, and then we'll talk about kind of the search for therapies. So maybe Fernanda, you could start just by describing, you living in the Bay Area, but obviously following this very closely in Brazil. How would you characterize the main differences that you see in terms of how COVID has impacted Brazilian society versus American society?
Yeah, so I think that we are all pretty much impacted in similar ways. And of course, we have our specificities. In Brazil, in March, in the beginning of March, we started to do some predictions, and even some data scientists, even from our group as well, where we could predict based on what we had in the other countries, what would be the peak and when would be the peak. And with pessimist and with some optimist scenarios and all the things, what actually helped the public, all the systems that we actually had the shutdown, and we saw that we specifically in Rio de Janeiro and in São Paulo was the center, and we saw that the peak that was was already predicted for April is now in 2 weeks. So Brazil is still going to see in the next 2 weeks the peak. So, but everybody, we have in the majority of the centers, we are all at home. Unfortunately, it's difficult to control. We have different levels of education in our populations, as you know. So in some centers, it didn't work as it should. But I think that the disease started much more with the middle level class, and now we see spread through all populations. Although São Paulo and Rio were most effectively, we have also heterogeneous distribution in terms of resource in other states. So we have different scenarios in terms of collapse number of beds in different states of Brazil. That's true, as you can imagine. And of course, we are a little bit 2 weeks earlier or a little bit more from what we had in US. Yeah.
Okay, so then let me take one of the things you said, and I'll ask both you and Nevin about this, because we've been hearing about these predictions, right? And how, I remember hearing that we were 2 weeks away from peak, maybe 4 weeks ago or 5 weeks ago. We all have our new routines, right, under coronavirus. Mine is I make my cup of coffee and then I go to this website that captures the new cases and the deaths from the day before. And one of the things that I notice, one is that in the US, we seem to have stabilized that, 25,000 to 30,000 new cases every day. The second one is, curiously, yesterday, Brazil jumped to number 2 in terms of like new cases, not cumulative and not deaths, but clearly people thought that Brazil was going to have it a little lighter, maybe because of the weather, etc. But that's not happening. So when you talk about 2 weeks away from the peak, can you tell me a little bit more about how confident you guys are in these estimates, given that things seem to be more difficult than maybe was originally expected?
Yeah, I think that, of course, this is all we are learning in real time, and all those predictions, they are also, the algorithms are also continuing to learn with the new, how can I say, the new scenario that we are having. So I think that if we don't have, and all the time, I think that if we have other interventions, these things can change. So I think that the way that they are predicting, the majority of them, they are actually giving a spread scenario of optimistic, a more realistic, and a more pessimist. But of course, also account with some interventions. So let's see. We were discussing if the politics and everything, that the seasonal were really having an effect or not. With the new situations, with the new cases, we know that we need that to strengthen this recommendation. That's what actually just came from Fiocruz today, actually, that they are just supporting the continuation and actually to strengthen the lockdown. Because otherwise, the collapse and the things that we are predicting the model as people were starting to go around and to be more relaxed, then we can actually have the model not working anymore. So I think that taking into account all the limits of the modalities and how we can really predict, I think they are doing actually a pretty good job, the majority of the models.
And what about you, Nevin? You know, given that you probably do a lot of work not only on quantitatively looking at proteins, I imagine you probably have some thoughts on these predictions of peaks. What are your thoughts?
I could give you my perspective on the trajectory of this virus in the Bay Area. So obviously this was a virus that we didn't know much about at Christmas. I remember reading about it at Christmas time. And then the first reported case was, I think in the United States, was in Seattle, and the genome was sequenced, and we downloaded that sequence, I think January the 24th, and we started to work on this virus with a couple of my top scientists. And then in about the middle of February here, I think it was the first community spread that was reported. That is to say, someone got infected and they didn't know how. They hadn't traveled to a place that was problematic or hadn't interacted with someone who was known to be infected. And at that point, it was the realization that this was everywhere and everything's going to get shut down. It did get shut down about March 15th in San Francisco. For us, we actually got a lot of work done in those few weeks before the shutdown happened. Scientists can still go to work, although it's a skeleton crew, only those that are deemed essential, i.e. those are the ones that are intimately involved in the COVID-19 research. But I remember that shutdown. It was March 15th, and the predictions were dire here in the Bay Area. They were saying we were going to be inundated, the hospitals were going to be inundated, it was going to be over capacity with the ICU. It was incredibly frightening. However, as time went by, it looked better and better. So we have these regular meetings at UCSF, these town halls, obviously via Zoom, where one of the top epidemiologists, George Rutherford, gives us updates. And every day it started to look better and better. And that was really due to the shelter in place, people adhering to the shelter in place very strongly. However, today he gave an update and there's starting to be a peak. And why? Well, because people are thinking it's not so bad. People are not adhering to these rules, and I can even see it in the streets. There's more and more people coming out, and that significantly frightens me. So people have to be vigilant. They have to stay indoors. They have to stay away from others, or we're going to see this peak. And you see history repeat itself. So if you look at the 1918 flu epidemic in San Francisco, initially they did a fantastic job keeping it under control. Then fall came around and they said, oh, we had it beat, and everybody resumed their lives. Well, they had an amazing peak in January. And I'm afraid that the same thing's going to happen here in San Francisco and maybe around the world as well. So I hope that doesn't happen, but I mean, people have to remain vigilant, and or we're going to start dealing with these problems, which are probably inevitable, not just here, but around the world.
Mm-hmm. So how do you, just before talking about your efforts, right, with regards to treatments and possible pathways that could be involved here, as a society, we've been grappling with this notion of like, economic impact versus human impact, right? And when is the right time to actually find that compromise? It seems like one of the advantages that many of the Asian countries had is that because they've had epidemics in recent history, once something is about to happen, I imagine they're quicker to actually jump to taking precautions, as opposed to the US, where we haven't had anything like this in recent memory. I imagine if we had another one in like 3 years, people would be a lot quicker to do social distancing and put on masks. But where are you on the finding that optimal point of not letting us have such a dramatic economic collapse that then it becomes really difficult to recover, versus not doing it too early to avoid, you know, the containment of the virus?
I mean, if you look, it's a great point. If you look at SARS-1, where the countries that had to deal with it, like Korea, Korea's one of the best countries in the world in terms of getting these things under control for SARS-2. And I think we should look at these countries that have dealt with it before and are dealing with it now much more successfully, and learn from that. And as you point out, the United States hasn't had to deal with this in 100 years, right? The 1918 flu epidemic is probably the best example. That's over 100 years ago. And this balance between the economy versus health, I mean, obviously our president is, let's say, pushing towards opening things up sooner than maybe some others are suggesting. And I mean, in my mind, it's a short-term, long-term thing. You're like, all right, let's get the economy going. But then longer term, you're going to suffer. So you have to be, in my opinion, you have to be vigilant short-term. Let's get rid of this. And then longer term, we'll be much better off economically, socially, and otherwise. But now's, in my opinion, not the time to relax our the shelter-in-place rules. It's now's the time to be even more vigilant, to make sure we can stop the spread of this virus. Because in long term, that's going to be better for us economically and socially and otherwise.
Mm-hmm. And then Fernanda, maybe I'd like to hear your answer on the same question. But with Brazil, it's a little bit more difficult, right? Because in the US, whereas this is a country that can actually print trillions of dollars in support for people's and salaries and companies, that's not the case in Brazil at all. So I imagine that the line has to be drawn in a slightly different place, right? But what are your thoughts on when is the right time, and what's the responsible way to approach that?
Yeah, you just said, we don't have the ideal situation, and we have to deal with the situation that we have. And I think that the impact for the low income class will be much higher. We know that's very difficult. We know that in many countries, although they were locked down, the governments were giving food, they were delivering all the support for the family, small countries. So we have a huge challenge. I think that we learned it also from US and from the other countries what worked, and we know, but we need to adapt it to our own reality. You're completely right. And this is something that even makes us a little bit more struggling even more with the how to draw the line, I guess. Yeah, you're right.
Yeah, we'll have to follow to see what happens and the repercussions. There's no great answer, right? So maybe we can turn to the efforts by both of your institutions in terms of, it sounds like treatments, therapeutics is the place where you guys are headed, as opposed to vaccines and diagnostics, right? Maybe, Fernanda, you can start talking about the efforts that your institute has, and then we'll pass it on to Nevin.
Yeah. So we have what we did as a little bit as we did with the Zika, but with a completely different proportion, because Zika was much more specific and touching 2 or 3 types of patients, and actually, but the COVID, as it's now we are seeing stroke, we are seeing it's not only even a disease, it's a systemic disease and impacting much more. So what we did was actually we really shifted, and all the institute to look for an effort to deal with the COVID-19. And we have some pillars, like we have a diagnostic, some efforts. We have some efforts with data science and epidemiology, and also with machine learning in terms of the clinical cases and spread through the hospitals. Of course, also clinical trials and treatments. nowadays what we're doing is much more using drugs that we know that we can use for different disorders, but now apply it to that and design all the clinical studies, and also communication. As a scientist, we need to communicate to the population and also to the health professionals. As you can imagine now, the papers, the articles, they are going spread to preprints, and many people that are in the front cannot really read all them. So we actually have a team that's taking care of reading and actually spreading that to health professionals as well. So the whole institute is actually dedicated to that. And what we are trying to do since the beginning is actually to collect structured data. Because we know that we need to give answers now. We learned with the other countries that puts Brazil in a really different spot. So we know now different questions that we need to target, but we need to collect structured data that will be able to actually to make us to give the answer for the moment now. But for sure, if we have structured data, if you have biological data, if you have a biobank, we for sure we will still learn with those with this epidemic. So that's how we organize ourselves.
Are you guys specifically monitoring what are the different efforts out there, different therapies that people are trying, so that you try it also in Brazil, or are you looking for additional therapies that could also be used? What kind of coordination, like what's the angle that you guys choose to prioritize?
Yeah, so there at IDOR, the majority of the clinical data is actually being collected and implemented with the very similar clinical trials that we have everywhere. Of course, now the anticoagulation and the targets are a little bit different, but to be able to use with patients now, we are actually using the proven drugs that are already doing, and some cell therapy as well. We are doing some stem cell trials also for stem cells from the bone marrow and also from cord. Those are also other therapies that we have ongoing studies as well. And in terms of the translational science with some cell models, then we are targeting different others, antivirus and others drugs, but then in human cells, but not directly in patients. So we have the translational target and the more what we can do right now for the patients that are in the hospital. So we have 50 hospitals spread through Brazil. So we have the registered in all those hospitals, plus 10 public hospitals in Brazil as well. So it's a huge, I mean, it's a big number of data that we are being able to collect, all of them unified.
Interesting. And how about you, Nevin? What can you tell us some of your efforts?
Sure. Thank you for that opportunity. As you point out, there's a number of ongoing efforts around the world with respect to COVID-19. There's diagnostics efforts, and then there's treatment, and there's vaccines, and then there are drug approaches and antibody approaches. So what we've taken is a drug compound approach, and we're taking a slightly different approach than others. A lot of other groups are trying to find drugs or compounds that would inhibit specific viral genes or proteins. We're not doing that. What we're trying to do is identify the human proteins that the virus needs in order to optimally infect our cells, and then through drug repurposing, as you just heard, identify drugs and compounds that would inhibit the human proteins that the virus needs to infect our cells. Obviously, these are drugs and compounds that are being used for other diseases, like neurodegenerative disease or cancer. So to facilitate this, we generated this protein-protein interaction map, looking at essentially all 30 of the viral genes that are connected to SARS-2, and identified the human proteins that are physically associated with the viral proteins in this map, right? So there's 30 genes in SARS-2, there's 20,000 genes in our cells. The virus can't live by itself. It needs our cells, our genes, our proteins in order to live and replicate and infect us. So what are those human proteins? So we identified over 300 human proteins, and then from that identified 69 different drugs and compounds that were known or thought to bind to one of these human proteins. And this is a mixture of FDA approved drugs in clinical trials, and then compounds being looked at preclinical. So there's advantages and disadvantages to this approach. Okay, so an advantage here is that you don't have to worry so much about resistance. You know, if you target a viral protein, you more easily get resistance, especially RNA viruses, because it mutates quickly. If you remember HIV, the big breakthrough was a combination of 3 drugs targeting 3 different HIV genes, and that's how you overcame resistance. Well, we don't have time here to get to these 3 drugs for the virus. So an advantage is targeting a human protein that the virus needs. Well, we don't mutate as fast as the virus, right? So you don't have to worry about resistance. Another advantage here is that we and others have seen over the years is that different viruses actually target similar human proteins. Okay, so the idea is if we had a host-directed therapy for COVID-19, it would be more likely to work for COVID-22, COVID-24, or other viral pandemics that come along that we don't even know about. So those are the advantages. A disadvantage, though, is toxicity, right? But if we're focusing on FDA-approved drugs or compounds in clinical trials that have passed the toxicity issue, they'd be less likely they'd be less likely to be toxic. Okay, so this is an approach that we're taking, host-directed, that we think it's pan-pathogenic. And during the course of this study, we've identified, as I said, less than 100 drugs or compounds that were looked at. We've looked at 2/3 right now, and about 10 are looking very interesting in a laboratory setting in terms of having potent antiviral activity. This is in comparison to other groups that are just wildly screening tens of thousands of drugs or compounds. I still think that's a valuable thing to do. We're taking a different approach. Ours is a data-driven drug discovery approach. We're identifying the biology first and using the biology to point us in certain therapeutic directions. Then if we see something, we can go back to the biology. And if you know the biological mechanism, you're so much more powerful in terms of tweaking the drug and the compound. And that gets you like 50 steps ahead in terms of novel pharmacology. So it's a host-directed approach, drug repurposing, data-driven, and letting the data tell us where to go with respect to drugs and compounds. And as I said, we've uncovered some very exciting sets of drugs and compounds that now we're trying to get better. We're trying to tweak these better since we know what the targets are. We're so much better off in terms of tweaking these and to get them more potent. And now we're trying to figure out how to get these into people via clinical trials.
And these clinical trials, are you doing them exclusively in UCSF, or do you have a larger network that is working with you on those trials?
Yeah, I mean, the good and bad thing, I guess, more the good thing about UCSF, there's not that many people infected with COVID-19. So I hope it stays that way. But we're interfacing with other groups around the world. For example, one of the drugs that we found is called Zotadafin. It's from a company called Effector, co-founders connected to UCSF. Davide Ruggiero and Kevin Shokat. They have a clinical trial ongoing right now with this drug in Australia for multiple myeloma. Okay, so now the question is, can we piggyback on this with respect to COVID-19? We're talking with scientists also and clinicians in Chicago and New York and in Europe as well. So it's a big web of connections, both scientifically and clinically, that we all need in order to combat this pandemic.
Yeah, and it sounds like you and Fernanda should also talk offline, because it sounds like she has a few hospitals available to actually try some of these.
I was just thinking that. That's a great connection here to make. Fantastic.
Yeah.
Well, look, I want to, just in the interest of time, I want to applaud you guys for your efforts, on behalf of, humanity. We are really grateful that there are people like you who are trying all of these different things to see if we can find a solution. Perhaps, if you don't mind, I'm just going to get a little lighter here and ask you to make a few predictions, and again, understand, none of this is official. You're purely speculating for the purpose of this audience. But do you think that we're going to find a vaccine, even if it just moderately reduces kind of mortality with COVID-19? When do you think that would happen if we are going to find one? They've never found a vaccine for coronavirus before, so this is going to be a humanity first. But are you confident that this can happen within 12 to 18 months, like many people are predicting?
You want me to? Oh, go ahead.
No, go ahead.
I'll be succinct here. So, as you point out, there hasn't been a vaccine for coronavirus, and there's been a lot of viruses there's no vaccine for, but there's never been so many scientists focused on one problem like this before. Okay, so my hope is that a vaccine will come. There's so many people looking at this. I mean, hopefully within a year and a half. I mean, this would be fantastic. But in the interim, what I think personally what hopefully will work even by the end of the year, there'll be certain drugs that'll become available. And I just like HIV, the cocktail, a combination of drugs was the big breakthrough. I believe that it's going to be the same case here with the coronavirus. Maybe remdesivir, it's looking a little promising, I guess, in clinical trials, combined with another drug or a couple of other drugs together. So if I were to be optimistic, I would hope that, there's going to be a decrease of infection and then probably a peak in the fall. I'm hoping in 6-7 months by the end of the year, there'll be a drug cocktail that maybe isn't the silver bullet, but that will be effective in hopefully a lot of cases.
I see. So your angle on this is you wouldn't want to put all your eggs on the vaccine basket, which a lot of people are placing. You're saying if we can actually have a good cocktail to treat infections once we have them, then that removes a little bit that desperate need for a vaccine.
I think, we should be pushing on all fronts as hard as we can across the board, because you don't know what's going to break loose. And something will come earlier, be effective, and then something later will be more effective.
Mm-hmm.
Yeah. I completely agree, and if I could complement a little bit, so I think I also hope, and I think that although it will be the first time ever, but at the number of efforts that has been put on that, so I believe that probably a vaccine will come. I completely agree that this is a learning curve. We are learning how to really treat better those patients, not only targeting the treatment of the virus, the infection itself, but also all the other consequences that we know that is making. So we know that now how we deal with the more severity case, what we need to add on those treatments, and as I told, I think that we are still learning, and the things will come, and I believe that we will learn how to treat better the infection itself, but also how to deal with those patients and to treat the systemic disease as well.
Okay, so then the last 3 little personal questions. When is the next time you're going to fly on a plane? When is the next time you're going to have a beer in a bar? And when is the next time you're going to go to a movie theater?
Well, you just talked about 2 of my favorite things, the first 2 things you said. Yeah, I mean, that's a great question. I travel a lot. Last year I was gone pretty much half of the 365 days. Now I go to the other extreme where I don't travel at all. Personally, I think I probably won't be on a plane in 2020. It'll be in 2021. But I hope I get a beer in me at a bar before the end of 2020. We'll put it that way.
How about you, Finn?
So I hope to be at least in an open bar soon, at least in a beer garden, something like that soon. And I also travel a lot. I go every month to Brazil usually, because I still am here in Bay Area, but I work there. Probably I will need to go, although now with this situation in Brazil, I think that we don't know how closed Brazil will be from the rest of the world. But I think that probably still I will have to fly in 2020, after June, probably, of course. But I really hope to be able to go to an open bar earlier than that.
Great. Well, listen, it was a real pleasure talking to you guys. I don't know, Margariz, if there's anything else you'd like to ask or say.
Well, we had a couple questions regarding how many people are being tested in Brazil, Fernanda. Do you know?
So actually, we know that that was already treated was very little, less than 0.2%, for example, but the government saying that they are gonna treat up to 22% of the population by September. But I mean, we don't know how and the structure, so it's a little bit tough to know what's gonna happen. However, as we had around the world and also in Brazil, we saw this month a lot of people getting together to build hospitals, to deliver respirators, to help the population, to build hospitals in Rio, for example, as you know. And also there are a lot of efforts to help to test the population. So a lot of people that are donating tests and a lot of people that are trying to develop intelligence behind also the test. So, but it's tough to understand how many people really right now is gonna to be test. Although the federal government is saying that they are gonna have enough tests by September to test 22%. It's tough to answer.
And Fred, just one quick question to Navan. So you mentioned that all of us should be highly vigilant and don't get together, don't socialize. So the governor of California is planning to open up the economy back in phases. So apparently most companies will remain closed up to the end of the month. So do you think it is too early? Do you think they should remain longer?
It's a very good question. I mean, so I can just talk about UCSF. I mean, it was supposed to be shelter in place until May 1st. Well, they pushed it back to June 1st. And then they're talking about certain stages. And personally, I'm struggling with what to tell the people that work for me. I mean, I'm tempted to say, you know what? You stay home another month. Let's just see how this rides out. And I know not everybody has that luxury, right? I mean, people have to get out there and work and make a living. But a lot of people, especially at UCSF, they can be still working from home. So it's a debate for me internally. What's the best strategy? And I probably will be a little bit more conservative on that front. And you know, these phases that Gavin Newsom, the governor, is talking about, they're very good, right? And we just have to monitor people, and they still have to adhere to these social distancing rules when they do get back into work. And we need more testing, because when you get a little outbreak, you got to be able to identify it immediately and contain it. So it bothers me a lot that we don't have this testing capabilities, the widespread testing capabilities, and that's why I'm a little bit, you know, concerned about kind of getting back to business. Can I just say one more thing? I know we've been talking about a lot of negative things, or just one positive thing, especially in the scientific world, that we've seen here. It's unprecedented, in my opinion, how fast things have happened. And that's really due to the collaborative nature of what's gone on. And in my opinion, there's a big problem with science. It's very siloed, very competitive. The infrastructure often penalizes those that want to collaborate, especially young scientists. But here you've seen so many silos being broken down. So many scientists have come together in such an unprecedented way. And to me, this has been a really great silver lining out of all this. It's clearly a huge tragedy, but I've seen how science can move, how fast it can move. And what I'd love to see is this infrastructure in place now to stay in place so that we're in a better position for the next pandemic. Or, you know, the question is, why aren't we working like this on all diseases in this truly collaborative international way? And, you know, connecting the Silicon Valley with Brazilians, this is fantastic. Why aren't we doing more of this normally? So I'd love to see this in place going forward. And I think it will be in place. And I think as scientists, we have to make sure that it gets in place, because we're so much better off in the biomedical community and how we serve the community in general.
Thank you. Fred, last thoughts?
Yeah, again, thank you very much for your efforts. It was a pleasure speaking with you. And again, hope you and everybody else can actually get that beer sometime soon.
Yeah.
And the beer is on me. I hope we'll get together for that indeed sooner than we all might think.
I won't forget that offer. I will not forget that offer.
All right. We'll keep in touch, Navan. Okay, well, Fernanda, Navan, Fred, thank you so much. This was, as you were saying, the silver line, people getting together. You just did that. This was a session because of this team. Thank you so much. And thank you, everybody that joined us here in the call, as well as on social media. This video will be available perhaps tonight or tomorrow. Thanks for joining Tête-a-Tech. We are continuing the session. Follow our channels and stay tuned for the next ones. Obrigada. Tchau.
Thank you, Margarida.
Thank you. Obrigada.